Hey guys, so welcome to day 3, where we will be discussing cell transport. So these are literally how things are transported across the cell membrane, which every living cell has. And the entire goal of cell transport is to maintain homeostasis. Homeostasis is just like a balance. The whole point of transporting different particles across the cell membrane is to make sure that I have the same amount of particles outside the cell as I do the inside. Hence the balance, hence homeostasis. The first thing that we need to talk about when we're talking about cell transport is the cell membrane. So I'm going to make a little note about the cell membrane. Alright. The cell membrane is made up of phospholipids. Remember, a phospholipid is a lipid, hence what is in its name. Alright, you have 2 layers of phospholipids, hence sometimes the cell membrane is also called the phospholipid bilayer. I'm going to write that down, phospholipid bilayer. Because it has 2, so bi meaning 2 layers of phospholipids, 1, 2. Remember, you have the polar side, which means that it really likes water. It's like, yes, give me more water. And then the inside section are non-polar. Okay, so these are the fatty acid sides. Lipids do not like water. This is like if you were to try to wash grease off of a pan and you were only using water, you would be extremely unsuccessful. Alright, that's why you have to add soap, because water alone is not going to mix with the grease on the pan because it's a lipid, it's fat. Alright, so nonpolar does not like water. They are not friends. Absolutely not. Okay. We call the cell membrane semi-permeable. So that means it only lets certain things in. So only lets certain things in. That's why whenever we've annotated a cell membrane, we always annotate it controls what enters, exits. The fancy way of saying controls what enters and exits is saying that it's semi-permeable. It's only letting certain things pass. It's like the gatekeeper, like the bouncer of the cell. So that's my whole spiel on the cell membrane. Now we get to go on the really fun stuff of the types of transport. So we have active transport and we have passive transport. So when we are talking about this, we know that anything that we're talking about passing is passing through the cell membrane, that it's passing from outside to inside of the cell. So active transport, I'm going to first go over a couple of characteristics. Active transport requires energy, or ATP. It goes from low to high, or we can say against the concentration gradient. Oh, I'm going over my line. Woo. Okay, against the concentration gradient. Gradient. Wow, I suck at spelling today. All right. If we think about a concentration gradient, I like to think of it like a slide. Okay, here's my little slide. Alright, if I'm going down the slide, that means that I'm just, I'm having fun. It doesn't really take a lot of energy to like go down a slide, right? However, if I go up it, like we've all tried to climb up a slide before, it just happens, right? That takes a lot more energy for us to be able to go against where we should be. That's exactly what active transport is. Now we have 3 main examples of active transport. The first one is called a protein pump. So that helps molecules go from an area of low concentration to high concentration, but you need a protein to help with that. The second is called endocytosis. So that's when the cell engulfs really large particles that it can't just, it's it's too large to actually go through the cell membrane, so it needs to go through a different process. And then the third one is exocytosis. So X like exit, exocytosis means that it's helping really large molecules exit the cell because they don't need them anymore. They could be like waste. Second, we have passive transport. So a couple characteristics. This does not require energy because it is going from high to low or down. You can also say with the concentration gradient, down or with the concentration gradient. Concentration gradient. I'm going to draw my little slide. It's so cute. Woo! Okay, little slide, and you would be going down the slide. Okay? We have a couple of examples. So the first one is diffusion. A diffusion is any particle that's going from a high concentration to a low concentration. The second is called facilitated diffusion. This means that it requires a protein. Now, a lot of the time you guys get confused on facilitated diffusion and protein pump. The difference, they both involve a protein. The biggest difference is the direction of the molecules. So a protein pump automatically requires energy. The protein is helping molecules go from low to high. In facilitated diffusion, the protein is helping molecules go from high to low. It's just the direction in which the protein is helping. Alright, the third is osmosis. So osmosis is specific to water. And remember, we have three conditions. You have isotonic, which water is moving out and in at the same rate. You have hypertonic, which means that you have more water leaving the cell than you do coming in, so that the cell shrinks. And then the last thing that you have is hypotonic. I like to think of hypo like hippo. That means that the cell gets larger because you have a lot more water coming in than you do coming out. So the cell is going to swell and then potentially burst. Alright, so you have all of this information that can help you annotate throughout your multiple choice questions. For example, anytime you see cell membrane, you can draw this picture literally. You could write phospholipid bilayer, you could write semi-permeable, you could write control what enters, exits. For active transport, you could write requires energy, goes against. You could write examples. So just some things to think about as you're going through and beautifully annotating your paper. Good luck, have fun, and let me or Miss Williams know if you have any questions.